receptors to target tumors, specifically Alexandria. We have witnessed a renaissance of this field over the last 10 years. And it has changed the paradigm of treatment of relapsed and refractory ALL. We were enthusiastic several years ago. This enthusiasm was dampened by some reality, development of antigen-negative disease and relapse, as outlined by Francesco yesterday. Touching quickly of this, the mechanisms of relapse have been multiple and they have been target as well as tumor-specific. One of the first described mechanisms is the splice variants, in which the molecules, the target molecule, in this case CD19, is expressed only in part and missing the domain to which the CAR T cells can dock and kill the leukemia cells. Other mechanisms have been loss of the antigen, like in CD22 targeted CAR T cell work. In this work from Terry Fry and Crystal Macker from the NCI then, which describes this phenomenon as a phenomenon that is not restricted to CD19. I've described yesterday antigen escape and the loss of the antigen in multiple solid tumors as well. Obviously, the researchers and the scientists have turned to alternative targets. Again, Terry Fry, who's been a leader in this field now in Colorado, looked at the TSLPR as a target molecule in leukemia. Other molecules have been the CD133, as is described by Monica now, which is a myeloid lineage target, as well as CD22. We joined this because we've worked for about 15 years making multiple specificity T-cell platform by mixing cells, by co-grafting CAR T cells with multiple CARs with distinct specificities, and also by making CAR molecules that can recognize more than one CAR, more than one target antigen at the same time. So looking back, we just considered multiple targets here, 20, 22, 40, and 52 as alternatives to CD19 targeting that could act as salvage, but maybe also as fallback. That may be proved in the clinic to prevent CD19 escape. That stood out were CD20 and 22 because they overlap with and maybe had more restricted expression in the ontogeny of T cells and had been clinically targeted before with no intolerable toxicities. Specifically, these targets, 20 and 22, including 19, have been targeted with naked antibodies, conjugated antibodies, as described now by Monica, as well as biallelic monoclonal antibodies that bridge CD3, which is expressed by effector T cells, and B lymphoblast. By doing this, recruiting T cells in a cytotoxic manner. We're interested and have had deep expertise in chimeric antigen receptors, and therefore our thought was to use this in our studies. First, we started to look at primary ALL by collaborating with the JAM and finding that primary ALL of B lineage here has expressed consistently very high levels of CD19, as very well known to you. But we found variable levels of CD20 and yet also variable levels of CD22, depicted here in this Venn diagram. We then looked a little bit more deeper into a bigger series of relapsed ALLs, post-chemotherapy, and this series of 12 patients you can see by focusing on CD19 and 20 positive as well, and 19 and 22 positive, a very high variability of the expression, especially with CD20. When we looked after CD19-directed immunotherapy in 5 patients here, using CAR T cells as well as antibody, directed antibody, you can see here almost complete loss of CD19, except for one patient to the far right, patient 20R, again with variable expression, but preservation of CD20 and we had the opportunity to look at two very special patients who had CD19 negative escape after CAR T cell therapy. 21R is a patient to the left, and 22R, as you can see, before the treatment, typical expression of 19, 20, and 22. It is also variable for expression of CD20, and after being subjected to CD19 targeted therapy, complete loss of CD19, but also very interestingly, decrease of CD20 and 22. So at this time we thought maybe we should not only target 20 or 22, perhaps it would be more reasonable to target 20 and 22, especially that this could bring in more mature lineages, more mature leukemia and lymphoma types. Before we did this, we validated the data. We got very interesting this phenomenon, particularly the depression of CD20 and 22 level upon the elimination of CD19 by targeted therapy. So we used CRISPR-Cas technology. CRISPR technology is an endonuclease that is able to be targeted to particular areas of the DNA, and in this we particularly directed it to a CD19 gene, disrupted this gene, and caused a complete 19 knockout. You can see here in red the loss of CD19 was associated with some decrease in 20 and 22, but these were also maintained at a targetable level. See here in a primary ALL patient, and also we validated this in a leukemia cell line called Raji. So collectively, what we found here could support our hypothesis that targeting 19, 20, and 22 could overcome 19 escape and could also result in enhancement of the anti-tumor efficacy. We use in our group in contrast to the majority of the centers, we don't use a lentiviral system, and we do not have to apheresis the patients. We collect the blood from the peripheral vein of the patients, about 90 cc's, typically 3 cc's per kilogram. In children, after the collection, T cells are processed, are activated, and once activated, typically using C3 and 28, we put them in cytokines, IL-7 and 15, homeostatic cytokines, and introduce the CAR gene in a retroviral system, which allows us to use much smaller volumes before we characterize these cells, expand them, and then reinfuse them into the patient. We try to do our experiments in a very clinical translation conscious manner, so that in the future we will not have obstacles of having to redo the system. The typical product would be a trivalent CAR. We call it TriCAR for simplicity. Here, targeting 19, 20, and 22 using separate CAR molecules. This is the experimental design. We decided first to design and test the efficacy of the product, and then test the efficacy against a relapsed CD19 positive ALL, as well as efficacy in relapsed CD19 negative escape leukemia that lacks the CD19 target. First question we asked, can we produce tri-specific CAR T cells that maintain stable expression of all 3 CAR molecules distinctly? Again, we would like to do this in a clinically viable manner. Introducing 3 genes is probably not very viable by the FDA and other regulatory agencies. We use the 19, 20, and 22 engaging exodomains for the CAR, the transmembrane domains, and the zeta chain, which I showed yesterday, which is the initial signal one, a turnkey for the T cell. In addition, we used the 4-1BB as a co-stimulatory molecule, as shown here, and we put this in one cassette, one gene that has all 3 molecules, DNA molecules, and separated by a 2A sequence where it can be cleaved into 3 separate molecules that would surface on the surface of the T cell distinctly. Are they expressed on the surface? To the left is a single CAR T cell, a typical CD19 CAR T cell expression. To the right is our cell that expresses 19, 20, and 22 CAR on the surface of the cell. When we characterize these were predominantly, as shown here, CD8 cytotoxic cells with a minority of CD4 helper cells, and in regard to centrality, the majority were effector and central memory, and they were very comparable to the CD19 CAR T cell production. Testing more on the efficacy, first we started with primary leukemia that expresses CD19, asked the question, are CAR T cells efficacious against primary B-ALL? First, we developed a tool. A brain tumor cell line that was force expressed to express 19, 20, and 22. So we could control the 19, 20, and 22 expression. And showed here in this assay. This assay is called the excelligence assay. It is basically a confluence of target cells, in this case the OI medulloblastoma cells, that force express 19. And the growth of the cell here is maintained in this horizontal line, and when the cells are killed by the CAR T cells that are added here, the cells go to the bottom line, denoting depletion of the cells in a cell index that has some calculation. In this case, we found that CD19 positive DOIs were killed by the CD19 T cells, as shown here in black, and the trivalent CAR T cells targeting CD19 and 20 and 22. But when we expressed the 20 on the OA cells, only the trivalent CAR T cell was able to kill it, but not the CD19 CAR T cells. And similarly, the 22 was only distinctly killed by the trivalent product. Next question was, are these cells efficacious against primary ALL with different antigen profiles? We used these brain tumor cells that express the leukemia antigens in different values, in different ratios. Very dense 19, less 20 and 22, very dense 19, less 20 and less 22 to simulate some of the patient lines, and then another variable here. And consistently we found that the trivalent CAR T cells caused more lysis, more killing of the target cells shown in the red bars, compared, and this was significant in all conditions compared to the CD19 targeting CAR T cells. What we found that was very interesting is that when we tested the proliferation of the T cells, these cells only proliferated comparable to the 19 CAR T cells, which denoted that this effect was not because cells proliferated and increased in number, thereby have a collective better activity. This perhaps denoted that individual CAR T cells on the trivalent side are better killers, like any army perhaps has better single fighters. So we started looking at Hisham's, and fortunately he had the flow cytometry core and cell characterization. We asked questions, can we observe morphologic differences on the CAR immune synapse? And the immune synapse is the first touch point where the CAR T cell touches its target and starts the process of killing the target. So in this experiment, researchers in Hisham's group looked at the doublet cells, which denoted two cells or more sticking together, and then they looked at the cells of B cell and T cell origin that are stuck together using distinct antigens for CD3 and B cell antigens simultaneously, and then took this doublets and looked at the interface that can characterize the DNA of the T cell as well as the DNA of the target B cell. Lastly, we stained for the DNA, stained for the phalloidin, which is a stain for the cytoskeleton, and we looked at this immune synapse. Again, this is the contact point between both cells, specifically in this area as denoted here. What we found is that the trivalent CAR T cells exhibit higher levels in terms of number, density, as well as size, shown here in red, compared to the CD19 CAR. So they exhibit bigger, thicker, and deeper T cell contacts, which denotes perhaps better killing efficiency. Hisham also looked at a deeper characterization by comparing the distance between the B cell nucleus and the T cell nucleus. This has been characterized, especially in NK cells, as an indicator of the efficiency and the strength of the killing synapse. By comparing these, there seemed to be a advanced tricar ratio between the nuclei of the B cell and T cell, and this denotes the maturity, higher maturity of the synapse and higher killing potential. By attracting the microtubular organizing center to the synapse, this denotes better killing. So we could say that the trivalent CAR T cells are able to have a better punch, a better contact point with the target. How about, does this affect the behavior of the trivalent CAR T cells? We collaborated with Nabil in the University of Houston, which are our neighbors, and looked at a very interesting assay. Nabil is an engineer, he's not a biologist, but he invented a plate that has 10,000 wells and a method to dispense cells at specific ratios. So we could put a 1-to-1 cell, T cell to B cell. We can put one T cell to 2 or 3 or more targets. And we can study the time they take to seek each other, the time to contact, the time they stay in contact, and the time that the T cell needs to kill its target and also to kill its subsequent targets. This we call in this specialty serial killing. Just to envision this is a small video of a CAR T cell shown here in this blue color, this cell here, and a tumor cell shown in this color, and there is a reported dye, an XN5, which denotes that the target is being killed. When you look at this video, the T cell is seeking its target, then it gets in contact first with one of them, and then turns it into the NXN5 dead, then seeks the other one, turns it into the yellowish color NXN5 dead. You can multiply this data by about C19 CAR T cells, and the tri-CAR T cells are engaging Raji in co-culture and Raji knockout. We found significantly higher polyfunctionality T19 CAR T cells when 19 is knocked out. There is exclusive activity and polyfunctionality from the trivalent CAR T cells. On the other hand, when we looked at the profile of these cytokines and defined what compartment these are in, whether effector, chemoattractant, or regulatory, there was significantly more effector in the effector compartment of trivalent CAR T cells upon encountering the Raji, and upon encountering the knockout, there is only exclusive presence of these killer cells. We took this to test it in mice and engrafted these mice with in a CD19 knockout Raji cell, and showed here that there is control only by the trivalent CAR T cell, although the 19 cells are efficacious against CD19 positive disease. Here they have no more efficacy above the non-transduced or control cells. We repeated this in primary ALL, that we have knocked out CD19, and you can see here substantial and significant improvement over the killing by 19 CAR T cells that overlapped with the non-transduced T cells and were inefficacious. So in summary, what Dr. Hisham Azim's lab and mine have shown you that the efficacy against primary CAR T cells seems to be significantly higher in vitro against primary ALL. There is improved synapse and improved killing behavior of trivalent CAR T cells, especially with primary as well as C19 loss CAR T cells. The tri-CAR T cells are exclusively able, but not the conventional CD19, to kill primary ALL that has lost CD19 because of escape or knockout using gene modification. And lastly, we tested this in vivo in the absence of CD19 and showed distinct efficacy of the trivalent CAR T cells that co-targets 20 and 22 compared to CD19 in the face of CD19 escape. So, in conclusion, this again states my previous statement. Are they better suited to salvage or upfront? These are questions that need to be addressed. Typically, these questions are clinical trial questions. A very important question. Will this result in less antigen escape? Will we observe the same degree of antigen escape that is looking much worse than we thought it would be? After on the 3rd to 5th years of CAR T cell therapy, we're seeing 50% loss of the complete remission. So patients relapse in about 50%, and some of the series even more than 50%. What is influences escape? We have some evidence that it's an epigenetic phenomenon. And lastly, we hope to answer these questions, but in parallel we're developing an investigational new drug application for the FDA to test this clinically. I will not take time to thank the group. I'll now take time to thank groups. First and foremost are the patients and their families who volunteer to do this work. Hisham's group, my group, and the Center for Cell and Gene Therapy, and the funding agencies, the logos of which are there. Typically, predominantly this was a William Lawrence and Blanche Hughes Foundation funded project. Thank you very much.
So the floor is open if there's any questions.
Nabil, thanks for this very advanced. You took us to the other side of the world about the advances in the CAR T cell in LL. And you asked in the last slide, maybe CAR T cell or CAR T cell can overcome the antigen escape with the CD19 negative later on. Do you think also that CAR T cell is still effective to prevent relapse with CD19 CAR positive also?
So this is a very important question. I'll invite Hisham also to answer because he's the T-cell biologist. I don't think we can in this system answer this question because of the heterogeneity and different behavior. I would anticipate that we need to use tens of patient lines with heterogeneous expression in order to derive a rate of relapse comparing 19 and the trivalent product.
So your question is really, can this prevent CD19 positive relapse?
Yes.
So this is really has to be tested in the context of a clinical trial, but the CD19 positive relapse is mainly to lack of persistence of the CAR T cells, and it's unknown, at least in this system, how the persistence of the tri-CAR will basically compare to the standard, which is a single CAR. This is the first question. The second question, there are very rare cases of relapse with CD19 positive disease. Still the CAR T cell persists and still there is no killing, and then it's inferred to either a specific mutation in the X domain in the CD19 positive CAR, or transduction of the CAR T cell themselves with CD19 to do CD19 expression, or exhaustion of the CAR T cells that they don't function, although they are there. And again, all of those are possible questions. What would be hard to answer them outside the context of a clinical trial. But at least based on the preliminary data that we have here in the in vitro system, that does not seem to be the case. But again, ultimately the answer will have to be through a clinical trial.
Thank you.
Okay, Dr. Nabil and Dr. Hisham, thanks for the amazing presentations. Maybe I'm trying to oversimplify things, but the core essence of the whole thing you're trying to.
And Dr. Monica Metzger. Our dear professors Nabil, Ahmed, and Dr. Hisham, both of you don't want to remember, don't want, you will always remember Egypt, but give you a small souvenir as well. And of course, the great effort done by Ahmed. If there was no Ahmed, then there was no Last but not least, I don't know how to say it, but for this guy, if he was not here, I was not able to do many things, not this conference, but many things. So please give a great applause to Ahmed Nabil. And I would like to thank as well Dr. Ahmed Sedhun, Dr. Hafiz, Dr. Ahmed Ashur, who also did a great job for the presentation. And thank you very much. And we hope to see you next year, 5th and 6th March next year. We don't know if we'll stay here. Probably we'll stay here, but we could discussing if we go to another city in Egypt. Thank you very much and hope to see you next year.